So we are about to start the second session of the day. It's going to be a long day, but I'm pretty confident it's going to be very engaging. So the second topic is multimodal networks and integration, and I believe Sushmita is the one leading this effort.
All right. Guess I can put this down. Can people hear me? Okay. Can you hear me now?
Yep.
Okay, good. Let me just make sure I know how to navigate this. Okay. And then most likely I want to.
Just for laser pointer, you need it underneath where your
B?
index finger is.
Oh, okay, B. Okay. Okay, all right. Hello, everybody. I just wanted to echo what Anna said. This is such a great opportunity to meet all of the people who've been thinking about this problem. So very excited to be here. So our section is multimodal networks and integration. And I wanted to start off with a disclaimer that we were planning this to be after network inference. But hopefully, because it's a closed community, it would be OK. So we have organized this material. So this is being presented by Tony Gitter, Alex Pico, Sarah Goslin, Meng Jiang, and Hang Tang. And we'll be basically doing a tag team where we'll be presenting some material. And then towards the end, we'll have a concluding slides, and then we'll have a discussion. So I'll give a little bit of an introduction, and then just for the other participants of this session, like people who are talking, Sarah is going to watch us on the time, and if she's standing up, this means you just have 1 minute left because we have quite a bit of material. All right, so let's get started. So we thought we want to just maybe introduce what we mean by multimodal networks. So as we know, biomedical data is of many different types, and this can be genomic data, this can be imaging data, this can be network data, and these data sets are becoming increasingly available through a lot of systematic efforts from consortia and whatnot. So trying to leverage all of these data sets to get an understanding of molecular systems or living systems is really a driving question right now. So what do we mean by multimodal networks? And there can be different types of multimodal networks. You can have a situation where, now I'm going to use a laser pointer, where you have a node. You can have a node with, where did the pointer go? Oh, I see, okay. The B doesn't have any functionality, okay. So you can have a node, you can have one graph with one node type, but each node can have different attributes. And then you can have a situation where you have the same node type, but you can have different types of edges. And you can have also where diverse types of node types and diverse types of edges. And here are some examples of multimodal networks. For example, from multi-omics, you can have all of these nodes can represent different types of molecular entities like genes, proteins, metabolites, lipids. And then for each of them, you can have some different modalities. And then another type of multimodal network may be patient networks, where in addition to patient-specific omic profiles, you may measure other things like imaging readouts, electronic health records, and whatnot. So multimodal network data analysis methods, when we were thinking about putting the material, we thought the kinds of problems that they are trying to solve, so here are some of the things that you'll hear about. One question is, how do we take different types of data and try to reason, try to integrate and interpret all of these types of data sets? So integration of multimodal data using networks as a framework will be an important question to think about. The second question is the lack of systematic measurements of complete data sets. So these data sets, by definition, tend to be sparse and noisy. So how do we deal with the unknowns in these data sets? And then how do we go and integrate data sets that were not potentially, or measurements or information that was not measured as part of a study? This is where knowledge graphs can be really useful, and so we learn a little bit about that. And then finally, all of us are all excited about how do we build mechanistic predictive models, and then we'll have some discussions about that. So this is basically how our material is organized. We'll first talk about the network alignment problem, which you can think about is suitable to try to integrate different types of data sets, different types of networks. And then we'll talk about how multimodal networks, networks can be used to integrate different types of multimodal networks, multimodal data sets, and then inference of multimodal networks, and then knowledge graphs, and then finally we'll have some discussions. So with that, Hang, if you want to get started.
All right. So first of all, I want to thank Sushmita for her tremendous effort for coordinating this topic. So I will talk about network alignment. A few words about me myself. My name is Hang Tong. I'm a current associate professor at the University of Illinois. So my group does data mining for graphs and the network. We are interested in a variety of applications where very often graph mining can play an important role. And a large portion of my work is on the algorithm aspect of graph mining. So we do things like querying, detection, prediction, inference, and control. And in addition, we are also interested in the theoretical aspect as well as some system-related work of graph mining. So today I want to talk about network alignment, where we want to find the node correspondence across different networks. Now I should mention that this work was largely done not in the context of biology, but rather in the recent DARPA project on modeling adversarial activities. But I believe that applications of network alignment goes beyond adversarial activities, including biology, and also in finance, in social networks, and so on. So here's the plan. I will briefly show you some recent work we have done for network alignment. I will try to keep it at a high level. And then I want to share some high-level thoughts about some possible things we can do with network alignment in the future. Okay? So very roughly speaking, in network alignment, given the multiple networks, we essentially want to construct a tree of networks where each network lives in certain location of this tree, and then different networks, they are interconnected with each other, if you will, by the branches of this tree. Okay? So the general question we want to answer is how can we distill the value, such as the knowledge, from the multiple networks? And our solution is to align them together by addressing the 4 V's characteristics of the real-world networks, including the veracity, variety, volume, and velocity, which we refer to as the big network alignment. So this is an overview of some work we have done for big network alignment. I will not go into the specific algorithm, but essentially we have tried 2 types of methods for network alignment. One is based on optimization, where we essentially view one network as the noisy permutation of the other. So if we can recover this permutation matrix, we solve the network alignment problem. And then the second approach is based on the graph neural networks, where we first want to find the embeddings of the nodes of different graphs, and then the network alignment problem essentially becomes a point registration problem. So this is a more detailed overview of some recent network alignment algorithm. You can find the details in some recent tutorials we give at the CICAM and also at the KDD conference. So let me just give you one example on neural cross-network transformation. So in the 2 types of method I mentioned, the optimization-based method or the graph neural network-based method, both methods require some underlying assumption. For example, the optimization-based method either assume the linearity or the consistency, right? Like one network is the noisy permutation of the other. Now, as for the graph neural network-based method, they often assume that the embeddings of the nodes of the different graphs are in the same embedding space, or the embedding space of one graph is a linear transformation of the other. So in this work, we try to remove these assumptions, and we ask a more generic question. That is, given 2 arbitrary networks, Now, what will be the mathematically optimal way to transform one network to the other? So our solution is based on the encoder and the decoder architecture. Basically, the encoder will cosine the source network at the different granularities, and then the decoder will reconstruct the target network at the different resolutions. And during this process, we'll also identify the associations or the alignment between the source and the target networks at the different resolutions. Okay? So next, I will briefly share some thoughts about a few possible things we can do with network alignment, and I will focus on the following three questions, including when or where to align, how to align, and whether we want to do alignment at all. So first, when or where to align. So this is a very typical two-step procedures of network alignment, or generally for network integration or network fusion. So in this procedure, in the first step, we align the multi-source graph into what we call the worldview graph. And once you have this aligned graph, then we devise some data mining models to find the desired pattern. So here, our observation is that in the vast majority of the existing works, these two steps are often conducted separately. So a natural question is if we can combine these two steps together to construct or to align an optimal graph for some specific downstreaming data mining tasks, such as link prediction, classification, and so on. So next, how to align. So here, our main observation is that for the vast majority of the existing network alignment methods, they belong to what I call the additive alignment. So if we have 10 graphs, each with 1 million nodes, let's align them together to have a graph with 10 million nodes, right? Or if we have the embedding vector for each network, let's concatenate them together to have a longer embedding vector. So this type of additive alignment works well in many cases, but beyond that, how about what I call the compositive alignment? So what do I mean by this? Let me give you an example of the network of networks. So this is some early work we did in bioinformatics, where we are given the collection of the blue tissue-specific protein-to-protein interaction network. Now, in addition, we are also providing a green phenotype network. So in this case, instead of simply adding different blue tissue-specific PPI network together to have a bigger PPI network, we can instead compose these blue tissue-specific networks with the help of the phenotype network into a network of network model, where each green disease node itself will be mapped into another entire network. So this way, we might be able to recover the underlying hidden structures among different blue tissue-specific PPI networks. So finally, whether we want to do alignment at all. So here, our observation is that it seems that in the data mining community, there are two lines of the work, each pursuing a quite different idea, right? Including the one is on the merging idea, the other is on the splitting idea. So for the merging, the idea is that let's merge the input graphs, let's align them into the worldview graph, and then let's do the mining from there. Now, on the contrary, for the second idea, it pursues a splitting idea. Given the input graph, let's split them into smaller but semantically more meaningful units, such as the clusters, communities, motifs, and also the graphlet that was mentioned in the previous topic. So the natural question is, when should we pursue the merging idea and when should we pursue the splitting idea, right? And also, currently, these two lines of work are almost conducted orthogonally. So another interesting question could be if there is a sweet point where we can combine these two lines of work together. For example, maybe we want to first split them into smaller motifs or graphlets, and then let's align them together, but let's do the alignment on those semantically meaningful units on the motif graphlets level instead of the individual node level. So I think that's all. Next, I'll turn to Sarah and Tony.
to both time myself and you can time, well, we're doing 20 minutes between the two of us, and we're going to jump back. Okay, so, can do this. There we go. So the work that Tony and I are presenting really focuses on integrating multi-omics data from diverse experiments and using the graph as a tool to facilitate interpretability of that data. And it's really premised on this idea that omics data is inherently noisy. I can go into great detail about various aspects of noise. I give two visual examples here using this. There we go. So we know that RNA-seq counts can exhibit a bimodal distribution, and it's very hard to distinguish poorly expressed transcripts from those that are actually just noise. Proteomics data shown here below, which I didn't complete the citation for, apologize, also has a lot of missingness based on proteins that don't fly or those that don't, isoforms that aren't flying in all of the samples. I'll go into a lot more experiments about that. But there's a lot of missingness. And so I should always allude to the statistical tools that have been built to compensate for this. We have a lot of, and I just list them here for the purpose of completeness, but we can identify which transcripts, proteins, genes are interested in an experiment because of the phenomenal statisticians that have built these tools and made them reproducible. And then on the right here, I list tools that help us interpret the results of this. So we're given a set of proteins or genes or transcripts or metabolites, which ones are biologically relevant given the experiment that we've carried out. However, a lot of times these bag of genes techniques fail to really provide context for various biologists and really identify testable hypotheses. So the protein interaction network in particular, and I didn't allude to this because it's going to be covered more in network inference section, is using protein-protein interactions, particularly those from the literature, to really map biological measurements, whether they come from genes, transcripts, proteins, to an interaction network, which is fantastic. So here's an example of one such network, including my favorite protein, NF1, and using the physical interactions from the string database, and the edge width indicates sort of confidence, which is how much they're measured in physical experiments only. So this is what it looks like when you map a biological experiment to a protein-protein interaction network. The network is very dense, the measurements are sparse, and it's really, and so if you give this to a biologist, they'll just laugh at you and will never talk to you again. So I don't recommend doing that. So this slide ended up a little too small. I pulled this from a review that was written in 2013, at least the top, but I tried to sort of categorize these, and there's some mixing and matching, but there's this idea of network pruning versus module detection, which are sort of the most common algorithms that we like to use, that we're again taking omics data, mapping them to a network. And so I linked to them here, and they fall into this idea of network diffusion type algorithms that rely on one formulation where you're diffusing scores of, I don't have all the equations here, scores of nodes based on their presence in an experiment across a network. Community detection, which is used for data integration, which is a slightly different formulation of a similar idea where you're detecting, again, modules that behave together. And then there's this network pruning approach, which we'll talk a little bit further about, which are, the prize collecting Steiner tree, which is one major example that Tony will discuss. There's also max cut min flow, which again takes sort of two sets of nodes and tries to identify the paths between them. And then multi-commodity flow, which will map multiple experiments from these sort of pairwise experiments into a single graph. So with that, I will give it to Tony to sort of dive into the Steiner tree problem as our main example.
Thanks, Sarah. So we wanted to give one specific algorithm that can address this network pruning problem. Sometimes you might hear this called pathway reconstruction or subnetwork inference, but the idea is that we want to avoid this blue and yellow hairball that has all of our experimental interactions, all the edges, layered with the multi-omic data that's relevant to some specific condition, disease, whatever it is that we're studying. So this is not necessarily the best algorithm. Everyone has pros and cons. It's the one Sarah and I are closest to, so we can tell you all about it in great detail if you'd like. But plenty of other folks in this room work on other algorithms that have benefits. But let's just talk specifically about Steiner trees for a minute. So Steiner trees are similar to what we might want to solve with minimum spanning trees, but the big difference is that there's a subset of nodes that are of interest. So think about these nodes as coming from the multi-omic experiments. Maybe they come from a mass spectrometry experiment in a specific cell line, in a specific patient sample. So those are going to be colored here as the yellow nodes, and our goal is to find a way to use the graph, the protein interaction graph, to find connections among the yellow nodes. And I may say, again, use protein scores coming from experimental data, from these statistical analyses to decide what nodes are yellow or not. I can use edge weights from protein interaction databases, maybe like the string weights that Sarah mentioned, to come up with confidence scores on edges, and then translate those to costs where high confidence, more reliable edges are going to have very low costs. So the incentive is to try to find connections among these yellow proteins, yellow nodes, using high confidence, low cost edges. And essentially that's going to be our goal. So again, if everything's yellow, it's a minimum spanning tree. The Steiner tree variant is different because we have to choose which of these gray irrelevant nodes might be important for forming the connections to the yellow nodes. To make things just slightly more complicated, but also address the reality of experimental data, Sarah mentioned with mass spec a lot of single-cell RNA-seq. Other experimental technologies are noisy, so we might have false positive interactions. We might have borderline statistically significant changes that we might have hints of being relevant to some specific condition, but we're not sure. Those things might have low scores. On the other hand, we have false negatives, so proteins that are actually relevant in the process, but they weren't measured for whatever reason. So our goal is to come up with a formulation for this subnetwork inference problem that accounts for that. And to do that, we're going to not insist on connecting every yellow node, all these proteins of interest. We're going to give them some quantitative score, and then weigh that against our edge costs and say, well, let's grow the network using these low-cost edges until it's large enough. It captures enough of the high-scoring yellow nodes, but not insist on capturing or connecting all of them in whatever subnetwork we're going to return. So that trade-off is captured in the prize-collecting version of the Steiner tree problem, where we're now the protein scores on the yellow nodes are the prizes, and we want to try to find the connections that accumulate enough of the high-scoring nodes. And as we see on the right, sometimes we'll be willing to prune or exclude some of those low-scoring yellow nodes. So if we want to think about this in a little bit of detail, we have these two objective functions. We don't have to have a single connected graph, so we may say we want a forest structure that's a collection of tree-structured graphs. And our goal is to weigh these two terms. So we have one term that says, if you look at the vertices in the forest you return, you're going to be penalized every time you exclude some vertex that had a positive prize on it. So we're summing over all the vertices that were not picked up. And then we also have to pay a penalty for all the edge costs that we do include. So incentive to not have a network that's too large, to not use those unreliable high-cost edges. But we don't really know how to weigh these, so we need to have some tuning or trade-off parameters that will control the size of the forest networks we get back. And I'm not going to talk at all about how to solve these problems. It's an NP-complete problem. I'll just mention there's been work on that both in theoretical computer science, and that's been translated to practical network biology problems. So Sarah and I in Ernest Frankel's lab worked on this one tool, Omics integrator, that uses a message passing probabilistic model to come up with approximate solutions. But others, like the R package on the bottom, are actually more discrete. They're using clustering and minimum spanning trees, so all sorts of ways to come up with approximate solutions to this hard problem, which again have their own pros and cons.
Okay. So I did want to allude to this idea that there are other algorithms out there, and one of the approaches that, one of the roles that I play at my institution, which is a national lab, is trying to find the best network algorithm given the data set that we have. And so, network diffusion, as I mentioned, really has been, I did not include citations here, used to identify mutational complexes in cancer. Community detection algorithms have been remarkable at integrating diverse data types. Max cut min flow is really for mutational drivers of phenotype. And you can read the rest of the slide. I think the point is that there's no one graph algorithm that seems to be ideal, right? It really depends on the biological question at hand and in the data that you have. And so, Tony and I each kind of put up our projects that we're looking at right now because we're in a field of experts that might have solutions. And so for me, I'm really looking at spatial data right now. And the left is the field of spatial transcriptomics, which I think is really quite cutting edge, at least in cancer, where we're able to look at, RNA-seq from various regions and match them to various regions of a tumor, which are known to be heterogeneous with different, ostensibly, expression of transcripts that can be mapped to networks. And on the right, I show what we're doing in the proteomics space as well, where we're able to take, with a little less granularity because it's proteomics, but various regions of the same tissue and measure proteins, up to about 5,000 proteins per little square at this point, and map them to phenotype from the images themselves. And this is one of these cases where, and I know in the spatial transcriptomics space, I don't know if I've added a citation, where they've been able to say, okay, not only do we have network information from the protein interaction network, but we also have spatial context. So we can assume that the network in one area that's measured is more closely related to something that's directly next to it than something across on the other side of the image, right? That these signaling networks have some kind of spatial relationship. So trying to refine that spatial relationship in the networks, again, knowing that the data are noisy and that we have protein interaction networks are overly descriptive in many ways. How do we identify what networks are active in different regions of the tissue? Oh, I did include some citations. Good, go me. Okay, so this is what I'm seeing, at least in the spatial transcriptomics realm. And again, no one has done anything in spatial proteomics yet. But this idea that there are packages out there, that particularly this one in the middle here is called BaseSpace, which tries to borrow information from the protein measurements that they're getting in a particular, call them voxels, spot, I guess, is what they're called in spatial transcriptomics, to better infer the diverse heterogeneity of a tissue. So I really think that there's a lot of room to add networks to this field, and I'm kind of excited to see where it's going to go. Okay.
So my introduction almost assumed we had a single omic data type that was static, and I think we have at least tools to deal with that setting. So I want to talk about some other areas where maybe we need more algorithmic development. One, I think, opportunity for growth is to look at changes over time. So we could think about measuring protein phosphorylation changes, maybe with mass spectrometry, phosphoproteomics. And this might say, on very rapid timescales, even within seconds, we can have an existing protein population in a cell that's being not created or destroyed, not changing in abundance, but the phosphorylation state of certain phosphorylation sites on those proteins is changing, and that's leading to downstream activity in the cell. So we can measure these phosphorylation changes of many proteins. If we imagine we have a collection of different proteins here, shown by these different line graphs, the takeaway here is that some of these changes in protein phosphorylation are almost instantaneous. They're very, very fast. They happen early in some cellular response, and others are happening later. So intuitively, if we think about timing or measuring node activity times and combining that with graph algorithms, maybe not Steiner trees, but others that are looking at directed paths through a protein interaction network, how do these things help support or contradict each other? So if I propose that one of these early proteins is regulating directly a late protein, maybe that's the case. If I say it's the other way around, that's some sort of logical violation. So there's this information in the timing that should help inform what sort of paths I find in directed graphs that come from a protein interaction network. And I think that information has been underutilized, especially when we think about moving beyond those local changes of direct interactions, but saying, okay, we have maybe this protein upstream in a pathway is only activated at a late time point for some reason. So if I have this multi-edge path that ends up and proposes the late thing regulates the early thing, that's again a logical inconsistency. It must be something else regulating this early protein early. And there's very little work, I think, on really doing this type of temporal reasoning on graphs. If we think about how protein information, temporal information has been combined with protein networks, there's this really well-known study that uses dynamic Bayesian networks and uses protein-protein relationships or pathway edges as a network prior. That's more of a structure learning take on things. And there's other work, including some of our own, that is closer to the Steiner tree formulation where we have an existing graph and we want to do some reasoning on that graph. And folks have adopted max flow-based approaches. Some have looked at, again, just customized logical rules that try to basically do temporal reasoning like the sort of things that I mentioned, saying, well, it's unlikely that this is the regulatory path because we have late things happening before early things as we order nodes along a directed path in a graph. So there's some work here, but I feel like there's a lot more opportunity. And then if we finally actually turn to the multimodal presentation I was supposed to talk about, we can do a lot of different biological assays and measure different things about the same cells, the same samples. So sometimes those directly measure something related to proteins. Other times they might measure maybe transcriptional changes. And there are algorithms for integrating transcriptional information and projecting it back to proteins and saying, well, this is the transcription factor that is a protein and is active based on the transcriptional changes I see, or the kinase that is active based on the phosphorylation changes I see on known substrates. So we can get a lot of scores on proteins, and I don't think there's great ways to merge these scores in a sophisticated way, because if I think about what I've done in our applied projects when we have multi-omic data, it's kind of the most possible naive thing. So I did 4 types of assays. Let's add them up, or let's take the max. And if we contrast that about what I hope we're going to hear about later in the machine learning on graphs paper, you think about graph neural networks, they have these very customized, learned aggregation functions that are integrating information at a node, at a protein, and at neighboring proteins, and coming up with something that's really customized and sophisticated. And I just haven't seen those 2 worlds meet, so there could be opportunities for doing multimodal omic analyses like in the Steiner tree world, but maybe using learned ways to combine some of this information. So there you have it.
Yeah. We have a lot more. We wanted to save time for discussion.
I have my own timer. Okay, I have my own, so, yeah. Okay, so far what we've heard about is we have networks as a skeleton to try to interpret data, but in most situations we actually don't know what the network is. And we have lots and lots of types of measurements. So what I'm going to talk to you about is some work that's out there in terms of using different types of multi-omic data sets at the molecular level, so transcriptomics, proteomics, and then use that to try to reconstruct networks. So this is what I want to talk to you about. So we have different omic levels, and then we want to feed that in outcomes and network, where the nodes are different types of measurement types, and the edges are specific types of connections. So if you think about the different classes of methods that are out there, we can put them on a range of what we know about the networks that are already there in the network that we're trying to construct. So it's a little bit circular. So on the one end of the spectrum, we have methods based on just link prediction or classification, where we have a reasonable number of already known interactions, and we want to leverage that to predict new interactions. So our network is incomplete, so we try to infer some of those unknown interactions. And then on the other end of the spectrum are methods which don't assume at all anything about the structure of the graph, and then they try to reconstruct what the graph might be. So that falls into the realm of structure learning. So the first is the very simple framework of assuming to try to cast the link prediction or network inference problem as a classification, as a binary classification problem. So here, what we are assuming is we have a set of examples that we know are true interactions. And in most situations, we don't know what the non-interactions are. So we take the complement of the positive set, and we draw as many examples as there are in the positive set, and we train a classifier. And we basically say, okay, which is an interaction, which is not an interaction. So that's the simplest approach. But it's a very powerful approach. And by definition, it is integrative, because all of the features, all of this multimodal data becomes features in our data set. Okay, and there have been a lot of ways, a lot of powerful resources, for example, string, which Sarah referred to, which is really, a lot of us actually use this as a way to interpret our data. is using a binary classification framework. And there are also these other examples of some databases, but this is clearly in no way comprehensive. But the problem with these methods is that it doesn't assume anything about the fact that what we are predicting is really is on a network. So there is some graph topology that we might want to be able to leverage. So this is really paired data that we want to make predictions on. And so then the next class of methods that we can think about are these graph embedding based methods. And I'll only touch briefly on this because I know the other sessions are going to talk about it, especially the machine learning and network inference. And so what is this problem? So this is saying, let's assume that we have an incomplete graph. So we have some edges, and we have some node attributes. And these node attributes are what these multimodal omic data sets can be. And what we want to do is we want to predict the interactions between the nodes based on the node features and the edge features. Okay? And you can do this by using graph embedding. And you can talk about graph embedding can be done using matrix factorization based approaches as well as graph neural nets, which I'll not get into, but I'm sure the others will talk about. So here is a very interesting paper which tried to do this. So I didn't find a lot of examples doing this for multi-omic networks, but I think we can probably draw these connections. So this was a paper where they were trying to predict gene-disease interactions, and that is the matrix that they are trying to complete. And for each of the genes and the diseases, they had features, and these features can be gene expression data, it can be interaction network, and then they compress that into features, and then they use something called inductive matrix completion, where you can basically take the low dimensional projection and then parameterize it. So basically you take this little matrix and parameterize it, you decompose it into these lower dimensional matrices, and then you can, it turns out that this is like a bilinear problem, and then this z is what are the parameters, and then you have the x and the y that you're actually measuring from all of these different auxiliary features, and then you can use that to try to predict the interactions that you have not observed. So this was a case where you have one type of network, but you have lots of attributes for each of the nodes of the network. Going beyond that, we might want to think about if we have multiple types of networks. So here we have, this is from a review, which again was multimodal data, but again molecular networks were not as much talked about here. So you have multiple levels. So each of these could be, say, L1 is a protein interaction network, or a metabolic network, or a gene network. And what the idea here is, for each of them, we have some network, but we are trying to actually predict intermodal interactions. So between gene, so you have gene networks and you have disease networks. We can predict interaction between genes and diseases, or diseases and something else, individuals. And so the way you could do this is, again, you could take all these different matrices, and then you could embed them in some shared space using graph embedding based methods, matrix factorization, or neural nets. And then you would get a feature vector for each of the nodes, and then you could, again, feed them inside a supervised learning framework or inside a matrix completion type of framework, and then try to come up with what are the missing edges. So, and so then finally, the last type of methods is really not assuming at all much about the interactions that are there, and that falls into the realm of structured learning. The methods that are out there could be broadly grouped into two categories. Those where the multimodality is being used to inform the topology of a single node type structure. So let's say gene expression networks, and you're using different types of data sets to try to inform what the connections might be. And the other one is where you have different types of nodes and different types of interactions among them. So, so the first one is what I've said as prior-based methods, and the second one is like multi-type. Some of these names are actually what I came up with, but because it was hard to come up with a nice description, but if somebody has a better way to describe it, more than welcome to use that. So what are these prior-based methods? The general idea is we have one type of data that we are trying to describe, let's say gene expression data, and we have these auxiliary data sets which tell us something about what might be the potential interactions between two nodes in the network. And what these methods are trying to do is they're trying to reconstruct a network. And work in this field has moved in two directions. One is using these auxiliary data sets as priors, as graph priors. And then the other one is to try to infer something about that, measurements that we don't observe, such as protein activity levels, because many of these gene network inference methods are saying, let's use the mRNA level of the protein to tell us what the activity level of the transcription factor might be. But these methods are trying to estimate what the activity might be, and that can help in network inference. I'm not doing very well on time. Anyway, so there are two main classes of methods. One is based on whether you put a prior on the structure, whether you put a prior on the parameters. And here is one way to think about it. So you can think about the thing that you want to infer, which is the graph, as a high-dimensional random variable. And all of these auxiliary data sets can be as priors to inform the prior. And what we want to do is infer the graph given the data. And when we actually compare methods that use priors versus don't use priors, we see that, there is a performance gain between the methods that use priors versus not. And then the other side of the coin are where you have multiple node types and you want to maintain that identity. So multi-omic, you have protein measurements and RNA expressions, and you want to infer edges among them. And there are two classes of methods. One is a simple regression-based approach where you're saying, let's integrate these multi-omic data by predicting one level from the other level. And the other one is actually more probabilistic graphical model-based methods that are trying to learn different types of probability distributions. So this was something that we did where we tried to integrate mRNA with protein levels. Very briefly, what we said was, let's use the gene expression data, which is more diverse and more complete, to infer gene expression modules, and then do like a regularized regression approach to try to predict what the protein regulator might be for each gene expression module. And so that's helpful when you don't have exactly measured, exactly matched sample measurements. But going forward, what it is becoming increasingly common is to be able to measure matched multi-omic measurements, and then how can we actually model that? And there are methods based on what are called mixed graphical models, where you are explicitly modeling the distribution of a particular variable conditioned on the other variables. So you can use a mixture of continuous and discrete variables. And here are two examples. One was actually trying to infer something about a causal graph. So they're basically inferring directed graphs, and they're trying to solve these conditional probability distribution estimation problems. And the other one was using just a Bayesian network. I shouldn't say just a Bayesian network. They're using a Bayesian network, and they had all of these different types of measurements. So just to summarize, really, it depends upon how much known interactions you have. On the one hand, the classification link prediction methods are simple, but they require a lot of training data, whereas the other ones make less assumptions about what is known. I think I will just skip this and move on, because we will come back to some of these questions in the discussion. So let's move on to the next presenter. Alex.
Great, thanks, Sushmita. All right, now for something a little different. As opposed to, so this will take about 15 minutes, and then we'll have a few discussion slides. It should leave a good half hour for discussion before lunch. So I think we're doing good on timing. Great, thanks to my teammates for that. So I'm Alex, this is Meng, and we're gonna talk about something a little different. As opposed to data sourced multimodal networks, we're going to be talking about constructing a specific type of multimodal network called a knowledge graph, and we're going to be discussing a few examples of how these are composed from biomedical concepts from traditional databases or from the literature that are tied together by a common semantics and then some of their applications. Okay, great. So we've seen some examples of multimodal networks being used with omics data, and knowledge graphs can be used in a similar way, serving as a reference or as a as a framework for integrating data sets. These knowledge graphs are also commonly used in machine learning applications for inference or for drug discovery. Here's a title and link to a paper recently published where they pulled together 18 different databases to construct a purpose-built knowledge graph for predicting drug repurposing. And the nice thing about using a knowledge graph in this way is that each of those predictions come with a mechanistic model to help explain why the predictions were made. More generally, if you're interested in a direct lookup of, say, drugs and their protein targets, any sort of single hop query, you can use a traditional database, and knowledge graphs don't really gain you much. Where they really come in handy is for two or more, these multi-hop problems where you're going across multiple types of nodes or multiple types of edges through or more complicated graph structure. And these knowledge graphs often include, in addition to the direct edges that are sourced from databases, might include inferred edges, or you can perform your own inference. And a key aspect of knowledge graphs that you'll hear from Meng and I will be mentioning semantics a lot. And so tracking semantics are really important to be able to combine across multiple databases so that you know one source's gene is the same as another source's gene, for example. We've seen examples of using network analytics. There's also a whole area interested in reasoning or performing federated queries, which is just queries across multiple knowledge graphs. And again, that's enabled because of the conforming to common semantic standards. Okay, so what I'm going to do now is give a quick survey of 3 different active knowledge base projects. Hopefully you don't already know about all 3. And I picked these 3 because they all take a very different approach to making a knowledge graph. There's not just one way to make a knowledge graph. So this first one is the Spoke project. It's led by Sergio at UCSF, but really maintained by a whole team of collaborators. It began as a multimodal network called Heterogeneous that some of you might be familiar with, which had about 17 different types of nodes in it and connections pulled in from a handful of different databases. It's grown considerably over the last few years and now has many dozens of nodes and edge types and millions of instances of each of these. All of this information is in a single graph database, a Neo4j database. And then there's a collection of scripts that ingest content from databases and update on a nightly basis to harmonize all of those different databases, again, into a single model. There's a web GUI for accessing this knowledge base and programmatic access. To give you an example of usage, in collaboration with NASA, the Spoke network was used to help analyze some transcriptomics data from some spacefaring mice. So NASA had collected differential gene expression data on a set of mice that had taken a trip up to the International Space Station and back. And then using Spoke, they performed random walks through the Spoke network, starting from all the genes that were in the network in the knowledge graph, and just going in all the possible directions that you could go in the graph, and created a series of vectors. The dot product of those vectors and the gene expression data allowed them to arrive at interpretations of the data that these are example paths through the Spoke network, from genes to anatomy nodes to disease nodes to symptoms. And what they ended up focusing on are the connections from, again, mouse gene expression data, connecting ultimately to just 6 symptom nodes in the Spoke graph that are relevant to human astronauts. Okay, second example, Wikidata. So Spoke is a Neo4j database curated by a defined team of collaborators. Wikidata has taken a commons approach, so anyone can contribute to Wikidata. You can log into Wikidata right now and add new information, correct information, write a bot to periodically ingest new information into Wikidata. There's a self-assembled group of interested members who make a very public, transparent process of making decisions of how to model things together and what belongs in there and what doesn't, and you can participate in those groups as well. So everyone's familiar with Wikipedia. There's about 6 million articles there. You might not know that most of those Wikipedia articles have a Wikidata entry supporting them, a semantically defined knowledge graph entry that's much larger and covers a lot more content than is in Wikipedia. This covers geographies, peoples, everything that's in the encyclopedia. So there's just a small biomedical corner to Wikidata that was initially populated by GeneWiki and WikiPathways. Spoke is a Neo4j database. Wikidata is a Virtuoso graph database. So it's compiled into a single graph database. It has this web interface that I'll show you in a second, and programmatic access. So just to give you a sense of how this works, if you were to visit an article at Wikipedia, you could identify that there's a corresponding Wikidata entry where most of the content in that article is actually contained in the Wikidata database as semantic triples. So items, properties, and values, and the collection of those triples make this graph. So, of course, corresponding articles about the ACE protein, for example, in the biomedical corner of Wikidata, there's a corresponding Wikidata entry. And you can access this directly on the web or programmatically. Again, getting semantically defined statements about these proteins or other biomedical facts. Today there are dozens of node types and edge types in Wikidata in this biomedical corner, and lots of connections. So just to give an example, symptoms are linked to diseases, diseases linked to drugs, drugs linked to pathways. All that information is coming from multiple sources and are defined in Wikidata in a way that you can make those connections to these concepts and many, many more. All right, the third and final example I wanted to give is a project called BioThings Explorer. This is led by Andrew and Chun Lee at Scripps Research. And what they've done is instead of making a graph database, they're not using Neo4j or Virtuoso, they've instead just defined a smart API for traditional databases to register to. And so the way that works is Uniprot, for example, is registered in BioThings, and their existing API gets registered in a way that it defines inputs that take in NCBI genes, for example, and outputs that provide NCHI keys or Uniprot IDs, or what have you. And each of the databases, there's now over 200, have registered in a similar way, following the same semantic standards for defining their API in a common way at BioThings. And so what this enables is when someone makes a query to BioThings, that query is digested into a series of queries that then are sent out to each of the source databases, and the responses are returned and compiled by BioThings Explorer and returned as a single result. And so this can scale in a completely different way than a local graph database, which is interesting. And it's also making real-time queries each time you're making a query. And so there's no asynchronous update issues with the data with BioThings Explorer. There's a website interface for this and programmatic access. All right, that's my survey. I'm now going to hand it over to Meng, who's going to talk about constructing a knowledge graph from text.
Yeah, so I'm Meng. I'm an assistant professor at Notre Dame. I'm like a researcher in information processing. So yeah, thanks for talking about there are multiple ways that you can.
Can you stand in front of the mic, or you want this one? You want to?
Oh, this one. Okay, good.
Just the microphone. I stand in front of the mic.
Oh, sure. Yeah, I can stand on here. Yeah. So yeah, Alex just talked about there are multiple ways that you can integrate a variety of databases into one large knowledge graph. And you think about knowledge can be represented as concepts or entities and relationships. And the relationships can be represented as head, relation, and tail tuples. There are multiple kinds of relations that I can define to create that large knowledge graph. And another way, so these are like taking the databases as sources. Another way is that I can directly use a text as source. So the text can be biomedical, biological, literature, publications. And then you can use natural language processing to solve the problem. For example, information extraction techniques. There are at least two subtasks to create that knowledge graph. First of all, giving you a sentence, giving you a text, identify the boundaries of the name of the concepts, right? It's called name entity recognition or concept extraction. And secondly, that given the extracted concepts and pairs, then try to classify their relationship, identify their relation. If you have specified the relation schema, if you already define the types of relations you care about, then you can make it a classification problem. So that's how we can combine natural language processing, deep learning, especially deep learning has making these language models really, really efficient and effective. So we can use neural information extraction to create a large knowledge graph. At the same time, we may ask this question, will we be able to extract all the knowledge from any sentence in the biomedical, biological literature if we just represent the knowledge as head, relation, tail, tuples? So the question is, are facts enough? Taking the middle, this is a knowledge graph we created. So the middle layer, which is blue nodes, are the statement sentences in the papers. On the left, you probably will see some, the head and tail entities or concepts. They are connected by the relations, like increase or reduce. But in those sentences, they do not only have the facts or the claims, but also associated with the conditions. On the right, you can see the conditions, the means, the experiments that they use to observe this kind of facts, or the temperature values, many things, right? Could be conditions in the statement, and we argue that these conditions they play a very essential role in scientific statements, because without the condition that we scientists, precisely specify, some facts may no longer be valid. So we have to extract the conditions from the text. Then we introduce this kind of thing that's called, condition tuples, not just the fact tuples, but those condition tuples. And even for fact tuples, we say there could be some internal structures in the head and tail. They are not just entities or concepts. It could be the attributes of a concept or entity. For example, like here, the inhibition of the Corkum influx, right? That's an attribute. You can just say it's a concept. You want to separate them. You say the red thing is the attribute of the green thing, right? So then, given one sentence, we develop neural information extraction techniques to extract the fact tuples and the condition tuples at the same time. That can be more complete, kind of lossless, to represent the knowledge. So the novelty of this kind of approach, including we extract condition along with the facts, we identify attributes together with the concepts, we open the concept of vocabulary beyond the databases, because you can identify some concepts that are outside of the vocabulary of the databases. And we try to open the relation schema. We try to take any kind of predicates in the sentences, try to make it knowledge lossless. So the technologies we use are deep learning enhanced natural language processing techniques, and specifically it's called sequence labeling. I'm not going to do very detail, but yeah. So with that, you can do something for applications like literature search. Beyond, now most of the search engines, they just use keywords matching. You can match the words, you can match the keywords, you can match the phrases. So think about giving you two articles, you try to know their similarity or relevance. What if we compare them based on the facts, based on the conditions, based on the heads and tails and relations they talk about, right? So if we can do some extraction of this text, we can extract the graph for each article, and we can compare the articles on the graph based on the conditions and facts. So that's more like a knowledge-based comparison, knowledge-based relevance. So that can make this thing more intelligent to search the similar article. This schema we call the CTGA. C is say, given you the unstructured content, T is to extract the tuples, G is to make it a 3-layer knowledge graph, and lastly use A, which is the algorithm to find the similar articles based on the graphs. So I think Alex and I mostly covered how to integrate the knowledge data, large knowledge graph, where you can see integrating all this knowledge data, knowledge graph with omics data and the clinical data can be really, really useful. Yeah, it's so references, and it's welcome for Sushmita for the discussion. Thank you.
So I just wanted to conclude, and then I'm going to invite my team members to come and facilitate the discussion. So we sought out to look at these 4 main problems, and you heard about different approaches ranging from trying to use networks as a way to interpret data using things like standard trees, to network reconstruction, to knowledge graphs to try to leverage different omic type data sets with patient level data and so on. As well as natural language processing based methods, how they can be used to try to extract information to annotate these different types of knowledge graphs. I think one of the things that we are all excited about is how do we take all of these very rich data sets and get to what we think are mechanistic predictive models. So what I mean by that is really try to come to a place where the model can make predictions about unseen perturbations and try to tell us about like what might happen in the future. And so we listed a few things, and I think I'm going to stop and then ask my colleagues to come to the front. But basically, in order for us to really get to these mechanistic predictive models, and all of these models that you've talked about, and also in the first session, all of them can make a lot of predictions. How can we go to a list of predictions to something that is testable, hypotheses that is going to give us something that is biologically meaningful? So what do models need to be able to do? They need to be able to handle noisy, incomplete data, high-dimensional data. They need to be able to work with different types of contexts, and context can be space, time, different types of organisms. And then one way to maybe think about what might be a useful experiment to do is coming from active learning, where one could try to see whether we can actually come up with a cost function for experiments that might be, that we could use to try to say this is going to be the most informative experiment to do in the lab. And then another question is, can we really discriminate from correlation from causal models? And that's going to be really challenging. And then finally, we want our models to be able to compute. We want them to be able to take inputs and then produce some outputs. So there is a literature in the field of kinetic executable models that can be looked at to think about some of these problems. So with that, I'm going to invite the rest of the team members, and let's start having our discussion. If you guys just want to come to the front. That's okay, yeah.
I'll stand up here.
Yeah, let's
Questions?
yeah, questions for us.
I hope not start with a random question. Generally, if you're going to do machine learning on these huge multimodal networks, we assume a probability distribution on some data here, and data points are drawn from some distribution, and then maybe there's another distribution, some kind of output. Now, when we have everything here, I can't imagine now where's the probability distribution and on what? How do we formalize this into? What are the assumptions behind these integration systems? And I'm trying to understand, and I think it can be all kinds of things.
I have an initial fairly naive response because I've never done this myself, but the papers I've read that seem to be successful have a very purpose-driven approach where they either construct the knowledge base themselves from scratch with something in mind in order to make sure it conforms to the distributions that they're looking for, or you start with a pre-made one and filter it ahead of time to make sure it's going to have the shape and aspects you want. Yeah, I don't know if these are really just being used off the shelf blindly for precise modeling projects.
This is probably related. I thought maybe is this instance of a huge knowledge graph one instance of from some distribution, or do we know that from this? Where is it? Anyway.
Okay, so maybe I can add two. One immediate comes to my mind is those generative model-based methods of graph. Just because of the generative nature, they naturally underlying probability distribution assumption behind that. The other probably more related to my own work on the graph alignment. I see one line recent research is on the optimal graph transport. So there, I think the formulation is quite unique. You define two distributions, then the alignment problem becomes comparing the two probability distributions over the graph. Now, I'm not sure if there's one general approach for handling the multimodal method.
So I might have a rambling question or comment, suggestion, then question. So you heard about network alignment in social network domain. The field of, well, obviously in graph theory, there's graph matching that's existing for decades. But on the applied end, biological network alignment started way earlier than social network alignment. And I feel like the two communities are completely disconnected. And I'm using network alignment as just example. This whole, like we heard, Mom is in my department and I had no idea what he deals with biomedical knowledge graphs. I kind of had an idea, but I'm learning. How do we reconcile different communities working on related problems, even in the same domain, often without knowing or benefiting from each other's knowledge? So this is more, I didn't expect an answer, but more a discussion of how to get there. And this was one of the motivations I used to sell this workshop to NSF, as in we need to merge data mining, machine learning, and traditional computational biology communities. And this kind of links to also for us computational biology scientists who collaborate with biologists when it comes to mechanistic understanding and hypotheses. Biologists often have no idea what they're trying to test or what hypotheses are. Here's the data, tell me what we should test. And we spend so much time trying to, we can rank predictions, but getting to mechanistic understanding is that they can't verbalize what they are looking for. And that causes so much anxiety for computational scientists. So it's kind of two different aspects of merging knowledge across disciplines. Any thoughts?
So yeah, I'm going to pull from my hat where I worked at Sage Bio-Networks, and we did a lot of these sort of like meta questions of how to get people to, cross boundaries, silos, whatever jargon you want to use. But I think it really comes down to like, I like to think of it as a software engineer, like what are the users asking for? Like what is the problem we want to solve, right? And I think sometimes you get different, and I always ask biologists, like what question do you have? And then we'll figure out, because I think that there are different ways. And then, because there's lots of different ways to answer a specific question, right? You can go to the knowledge graph space, you can go to the graphs, and everyone has their hammer, right? That they want to, and they're just looking for a nail. So I think if we kind of can generate enough problems, they will actually, and questions, they will actually fall into these patterns naturally. And then we can start to see, okay, these questions, like the mechanistic modeling question, like that, I've seen, we've tried to use graphs, we've tried to use Boolean logic, we've tried to integrate those, deep learning, there was a proposal that came out of that, like everyone sort of, that is, there's a lot of different tools that we can throw at it, but I think it would be nice to sort of say, okay, given this question, these are the suite of tools, and how and then we can start to integrate within that question column, right?
So my experience is the question isn't precise, it's more of try to do, and I've been sent down the rabbit hole many times.
Yeah,
And I it's like, exploring, and we can get what are the testable hypotheses.
What can you see in the data? What can you see in the data? I avoid those. But yeah, I think you're right. I think you have to choose the right questions as well.
So, can I say something? So, I think one of the issues is, biology is hard, and I think that it really requires people from different disciplines to be really interested in the problem, and it also requires like time, dedication, and trainees. We have to train students to really think about that biology. They need to think about the biological problem. And those algorithms that Hang talked about, I mean, it would be very interesting to think about how we can, like, integrate multi-omic data sets and, try to come up with graphs and try to integrate that, and that would be super cool. But it's not like, you just take the data and apply the algorithm, out comes biology. It doesn't happen like that. 90% of the time, we are spending looking at our results and trying to figure out what the hell, what does it mean? So I think that, it requires time investment. Workshops like these are actually great. I'm so glad that, you actually did this. Hope this happens again.
Yeah, I think indeed, I want to echo Sushmi. I think exactly the events like this will help a lot. So I remember when we had the Zoom meeting with Tony and Sarah, we suddenly realized we are actually working on something very similar, right? So on the Steiner tree problem, although my work is completely outside the biology, I think the connection between the two sides are much tighter than we thought. And the other thing, I think also somehow echo what Sushmi said. I think the mentality is important. So if we computer science data mining folks say, oh, just give us the data, we'll run the algorithm for you, or the other way around, right? So where the tools, I can use the computation tools, just give me the tool and I will run it. I think if we have this kind of mentality, usually it doesn't work out very well, but really work together.
So I have an idea for identifying informative experiments they want to throw out and see what people think. So here, my goal is to identify a set of genes on which I want to do more experiments. I don't know what experiments are, and this goes back to what you said, Sophia. So when you're trying to infer these networks purely from data, there could be some genes for which we can identify the pairwise interactions fairly accurately. But could there not be groups of genes for which the data does not tell us what the pairwise interactions are? You really have to throw up your hands. So in a sense, they are like undirected hypergraphs. Right? There's a group of nodes. I can't figure out what the pairwise interactions are. So perhaps we could focus our attention on figuring out what these groups of nodes are, because that's where we should be doing the experiment more carefully in order to identify more specifically how they interact. Now, I don't know how to solve the problem, but I wonder if that might be a feasible way in which to try to identify groups of nodes for which we need to do more additional experiments.
So in other words, we could come up with the precise questions that the biologists could then go answer.
Right.
I like that.
There's nothing wrong with that.
I like that.
It's like having a biology core in a computer science department.
Yeah,
It's kind of the opposite.
Yeah.
I'll run with that in a different direction. When we worked with these pathway models and Steiner trees, I quickly found that even if we have willing collaborators, doing the experiment to follow up on a prediction is so open-ended. Yeah. And then you have a meeting, it's like, okay, well, are we doing Western blots or targeted mass spec, or we gotta tag it and follow localization? Because let's say I've solved all the network problems and I can say this is the really important node in your graph. That doesn't tell us what the protein's doing and how to look at it, how to even validate something at the level of a graph prediction. So that's something I've struggled with is, well, one, I don't know how to get to the right subset of nodes. I kind of feel like knowledge graphs might actually help because it probably generalizes things that we do in a low throughput manual way. But even that, trying to do active learning in some settings, it's more natural because what you're modeling is similar to the type of data you can generate more of if you just do more wet lab work. But sometimes it's so nuanced to figure out how to test the prediction that we just hit a wall there, and maybe try a lot of things. Sometimes that takes 4 or 5 years to figure out what that one node was doing. And how do you repeat that? How do you scale it up, even if you have access to a bio core in a CS department?
Yeah, no, I want to sort of chime into this, but steer it in a slightly towards one of the other questions up there as well. So I love this question about sort of how do we do follow-up in a really useful way, and I think you're right about cross-training as being a big piece of that. But I want to bring us back to this thing about context, right? Because I think the knowledge representations are a great way of getting at some of that, but I think that, all of what we're doing is about, in some sense, about context, right? You can't possibly think about representing. Yeah, I mean, we do a lot of graphs that don't represent context, but they're not as, really useful in a lot of pragmatic ways unless you can say something about when is this happening. And I would even say sort of how confident am I that this is happening, right? Even in the context that I know about. So how do we communicate about that, and how do we do better than, you know, do we want probabilities, do we want models that are like, because I think you could put that on every single representation we've talked about today. Just sort of, when does this happen, how often does it happen in everyone?
Can you elaborate on what you mean by context?
Yeah,
The
right,
when.
so that's the other question.
When, I think the easiest is to start with the when, right?
I want to know what species, what genotype,
Yeah, I
what
think-
model system. I mean,
Probably know what
Yeah.
species, right?
Yeah.
So that's something that frankly isn't in a lot of publications, as I'm sure you all know, but could be captured, because people do know what species they're talking about, but, you know, what is being done with that species? What disease are we talking about? Do we know?
Maybe tissue specificity or?
Yeah, do we know, are we looking at specific tissues or cell types under certain circumstances? What don't we know, right? So how do we even represent what context is? Yeah. And I think that's a hard question, and it's a hard question that spans disciplines, right? So that's something where often when you're talking to a collaborator from another field, you're not necessarily in the same place on that. And I don't even know how we get at that, but I'd love more how to represent it.
I mean, this is one of the things that I have used, at least the networks for it. I think the knowledge graphs really help, is that, you know, it provides a baseline, right? Like, this is everything that you can possibly imagine has been curated into something, right? And then from there we can glean, okay, this is the experiment we ran in our, you know, slightly, you know, off model and micro tissues of this one tumor, right? And so that we only saw it, and so then we can say, okay, this is how we can represent this in the context of this larger set, and maybe we can identify some tissue-specific
I mean,
phenomena.
one tumor, I think you could say pretty confidently, even then you have cell types, right?
But it's a model system, and the models are imperfect, and, you know, because we had some cytokines. I mean, there's always something that changes things, right? So, you know, I like to say, okay, this is the baseline, but I think trying to figure out what measurement we're looking at and what, because I think that's the other thing, is that when you have so much data, it's hard to figure out, this is more the deep one, what black box versus white box. Like, how do you interpret the results that you get?
Yeah, but like if you make a multi-leap inference, which is the point of these models,
Yeah.
can you do that, right? Does that make any sense at all?
There's no
And
distribution,
it's
yeah.
not clear that we're even capturing that in a way where we could ask that question.
That goes back to the thing about the distribution, of
Yeah.
what the probabilities are on your calls.
Yeah.
And it's this weird balance between, you know, the source of the data and capturing those conditions so that you can precisely say what it is. But sometimes you want that blurriness because you're trying to find something new that hasn't been measured yet that you want to make a prediction. And so that blurriness can sometimes be an advantage. And
Right,
that
and the
balance
question
is
is whether
case by
you're
case.
taking things down by, you know, from 50% to 20% or 50% to, you know, 1,000 of it.
Correct.
In terms of how much work you want to do to try and follow up.
Yeah.
There's two of you. Who wants to
Sure. I'm just maybe too excited about these topics. So actually, I don't want to ask too many questions. Maybe for some kind of question is, I was thinking about this network alignment, how this, you know, this seems like this graph representation could be, in fact, the right way to do it. The reason is, you know, when we think about added distance on strings, like string alignment, it seems like for DNA sequences and proteins, that is the alphabet on which changes happen. It seems like string algorithms are maybe the right way to think about it. In network alignment, nodes are not anymore from that constant alphabet. It seems like there's something really different here. And I don't know whether we can do it properly with, you know, regular combinatorial methods. Maybe it just, I'm wondering whether this network graph representation type of alignment actually brings something new to this problem, whether we are learning something about these cost functions that is different, you know, just your thoughts.
Yeah, I think that's a great question. I think there's several ways we can do one based on the optimization part of the method, right? So somehow, some early methods, including the ISO-RAN that was actually from the biology, that actually has a lot of connection to like the network alignment algorithms will develop outside the context of the network problem. So I do think there is a lot of potential opportunity that the two communities can work together. So I think that looking at the appropriate cost function, like what do we mean, like when we do the alignment, like how do we actually add it to one network so that after we do this kind of editing can match to the target net as well as possible. So that more or less what this neural cross network transformation will be is the underlying motivation behind. The other thing that somehow relates to what you talked in your presentation, in many cases we can convert the graph data into multiple sequences, like using the random walk. And then once you have the multiple random walk, essentially you have to convert the graph into sequence for the multiple sequence. So a lot of concepts there maybe will be helpful.
I just want to go back because I think, what I've been talking about, how to test, right? But, I'm going to, again, put my biologist hat on, if I may, and, because we think a lot about, I think that's a very important question, and we think a lot about how do we do experimental validations, and we strive to do experimental validations on every of our machine learning prediction papers. And I think number one is you just have to find like the match, right? So you have to find a biologist interested in the specific, question that the knowledge, I guess, for that pathway or for that disease. If they're interested in that, I think that would be the start. Because if they're not interested in that, it's just very hard to say, hey, since you do biology, can you test this for me? I say, well, I'm not really interested in that pathway, in that problem. And the other thing also is, it is hard, right, with this multi-leap inference, right? So let's say you find this gene. I can totally appreciate there's some fuzzy, allow some fuzziness in there, but if you say this gene somehow impact associated with this disease, right, directly from gene to phenotype, that is very hard to directly test. But if there is some sort of, so you have to go into a little bit of a molecular mechanism. If you somehow say, oh, that gene is regulating this pathway, and that pathway somehow modulates the transcriptome. I'm trying to multi-model, right? And then this, this gene expression pattern is associated with the disease. Like I think for the example that you want to experimentally test, again, for someone who has actually really think of this a lot and try to do this kind of test, because it's really hard to do this test, right? Often you test a number of them once work, you'll be very, very happy and you put that into your paper, right? So I think you want to do the hard work, even if you want to pick the best examples, and for those ones that you really have a much stronger. I think that would allow you to be easier to test, right? Then you can say, hey, let's test whether this protein really affects the signaling pathway. And if I affect the signaling pathway, does it change the transcriptome? I think even if you don't directly test with the disease association, that's already several validations that you have provided.
I think the important thing is to try to think about what kind of predictions the model is making, what is testable given the
but it goes back to the context. Like, in what context are we making the predictions, right?
Exactly.
So that's, we all have, maybe it's not detectable by mass spec, right? Maybe it's, maybe we can't measure the transcripts because they get degraded typically. Like, there's all these sort of other confounding factors. But focusing specifically on the context of the prediction is important.
But it also takes discussion, right? You don't have to go down there. You can talk to experts in that disease, right? And then you can say, hey, and then I think that it's really, you have to talk to about it, together work out a plan to validate from the basics and a little bit, steps up.
But it seems that the man's knowledge graph could answer such questions.
Yeah,
So it links all levels of information, and perhaps you want to add one other edge to connect this gene to disease, and that edge is the experiment, and then you, in your core, find a person to do that, perhaps experiment.
That's what I'm saying, yeah. I think these, it's very...
Probably can try the link prediction to knowledge graph to try something new. And I want to add something a bit different. That is, I don't directly work on Omics data, so I don't push biologists to do some testing. But I do with NLP, so I have been observing the biologists having tested. So you look at literature, you find you can extract the facts, the tuples from the literature. Then you can calculate, or just count the tuples, right? You can rank based on their frequency to know what facts, what kind of relationships that the biologists have been paying a lot of attention to. And sometimes I even find conflicting relationships. Once they increase, they also reduce. And you look at their statements, you find, oh, actually, they have different conditions. So that could be a way to know what the biologists may be interested in. But on the other side, you also have the metadata of the literature, so you know the year. You can figure out like the trends, the dynamics of the research in biology. But I don't directly use Omics data.
Teresa had you had a hand raised somewhere. Do you have a question?
Well, I was just thinking that the definition of context is very difficult, and I don't think we will ever be able to define because there will be always a new context. So I think what's important is to somehow make sure that our tools will be allowing to define how we think the context may change the situation, or what aspect of the data may be changed by the context. So for example, you have, say, stress, they will be, DNA will be stressed, will have changed some binding site, which are perhaps weaker, maybe more. But also, it would be nice to somehow make those knowledge-based networks more dynamic, where we can maybe think about methods that will be helping us to answer how the context may affect. We cannot have all possible contexts in the data.
So can we define like a general purpose kernel function on context, whatever we're calling it? You might know context is a gene expression state, that's probably simple, but what if it's like context is just some term I extracted from a sentence and then the gene expression state, or times and all these things that we've talked about. I'm curious like how we can know if whatever it is we're doing is similar or very different from this fact or this other experiment or this conclusion, and then actually probabilistically or systematically reason over those things.
No. So better tools to mind the mistake. That's what we try to do, and that's what biologists try to do. They just have a lot of rich knowledge bases that they got to connect the facts. So we need.
I just want to mention, even talking about context, biological experiments now is actually quite advanced. I just want to, it's very sophisticated. For example, even if you're talking about context, right? So you could infer phosphorylation, for example, right? One of the events is very important for the signaling. And it's very hard to do this directly, but then, you can generate, very easily can generate a phosphomimetic mutant. So that you can just test that, and if I use that, and then that's a surrogate, it's a very good surrogate for the phosphorylation context in the cell. So there's many different, such things, available.
I think Ben has a comment question.
I had a related but slightly different question/direction, which is network inference. So if I understand a lot of the things that were presented, like probabilistic graphical models, most of these models don't deal well with feedback or directed cycles, which are actually present in a lot of biological systems. And so I wonder, any of you have thoughts, because it kind of goes with the theme of temporal data or perturbation data. Like what sort of data would we need? Where are we in terms of inferring feedback in these models?
I mean, you mentioned two things, right? So there's perturbations and there's temporal data, and we could go down the route, I think, for either one of those single data sets. There's been specialized probabilistic graphical models to deal with both of those. Nested effects models are a couple decades old, and I threw up a term, dynamic Bayesian networks, at least, that's kind of a root of these models that do structure inference specifically for time series data, and there's been a thread of methods there.
But how much really is, what's your sort of take on the current state of getting feedback in signaling networks?
So DBMs have been used a lot. The problem is that, the temporal, too long DBMs, DBMs, dynamic Bayesian networks, can model cycles, feedbacks, but you need temporal information. So if you have nice high resolution temporal data, you can use dynamic Bayesian networks. But the problem is sample size, right? I mean, you don't have enough samples to be able to do that. The other side is to use, relax that. What is nice about Bayesian networks is that, you get a valid joint probability distribution. But what you could do is you could say, I don't really care whether there's a good joint probability distribution, then you can just learn something like dependency network, which allow you to basically like trying to predict things, predict one variable as a function of the other, and there you can have cycles. But to be honest, not a lot. Like, you know, from a formal point of view, I don't think I've seen a lot. There's some work from Karen Sachs, which I saw in Journal of Machine Learning Research. They were talking about like causal cycles, you know, causal models with cycles and stuff. I don't know where it went, and I don't know what Karen Sachs is doing. So yeah, I mean, I think those would be some of the interesting things to think about.
So the in-person workshop participants can continue discussion during lunch. I do want to give opportunity to online participants if they have any questions. Okay, so let's thank Sushmita and the team.